So today I'll be talking about combined rTMS and MRI studies in animal models. MRI studies in humans have considerably increased our knowledge regarding the potential mechanisms of rTMS. However, subject-related variables such as gender, age, exercise, diet, and genetic background might affect stimulation-induced effects of rTMS in both healthy individuals and patient populations. Animal models give us the opportunity to reduce variability in rTMS effects by controlling for many of these factors. For example, we can perform experiments on animals which are of the same gender and at the same time ensure that they have the same diet and are from the same genetic background. We can even pick female rats, which are genetically bred to show high anxiety, and study the effects of rTMS in them. Being able to understand the effects of rTMS in such populations will drive personalized rTMS treatments in humans. So because animals introduce considerably less variability in experiments, they have been useful in elucidating some of the mechanisms of rTMS. The benefit of animal studies is that it gives us the ability to perform invasive studies of molecular and genetic changes that are not possible ethically in humans. However, little is known about how these molecular and genetic mechanisms relate to clinical changes. This knowledge gap is further amplified by the non-overlapping approaches used in clinical and preclinical studies. So clinical studies are non-invasive using MRI-based techniques, as we have seen, while preclinical studies are mostly invasive and performed post-mortem. If MRI is used in rTMS studies in animals, post-mortem data can then be acquired from the same animals at the end of the study, so we can link the two findings. The use of the same detection tool in animal and human studies will increase translatability of animal studies, as MRI can act as a bridge between what we see in post-mortem animal studies and in non-invasive human studies. In terms of stimulation delivery, our lab is specialized in miniaturized rodent coils. As you can see on the image on the right, mapping the magnetic field of this coil, the custom-built round coil I use in my studies delivers about 13 millitesla of stimulation focally to one brain hemisphere. So this is what I'm going to be talking about today. First, we'll look at the effects of one stimulation session in healthy rats, so the immediate effects. And then we'll look at the cumulative effects of repeated stimulation in healthy rats. And then I'll talk about an animal model of depression and what changes we see using when we look at the brain using MRI. And then we'll look at the rTMS effects in this animal model. So first of all, our question was, can resting state functional MRI detect TMS-induced changes in healthy rat brains? So we did a small study in which stimulation was delivered at 4 different frequencies, and MRI data was acquired immediately before and after the stimulation. So this is what the resting state network looked like. So you'll see a lot of these kinds of images in my slide. So if that's a rat, and this is a rat brain, if you slice it this way, you get the top coronal image on the left. And if you slice it that way, you get the bottom axial image. So following stimulation, we see a lot of changes depending on the frequency that we use. I'm not going to go into the details, but for example, as you can see here, following 10 Hertz stimulation, there's a decrease in functional connectivity in the ipsilateral somatosensory cortex and motor cortex, but we don't see that following 1 Hertz. So that now that we know that we can actually detect the effects of TMS using MRI in animals, the next step for us was to look at whether repeated TMS can induce cumulative changes in the healthy rats, and also how long do these changes actually last for. So in this study, we acquired information about the baseline resting activity by scanning the animals at the baseline time point. The animals then received daily 10 minutes of low intensity rTMS on the right side of the brain at either 10 Hertz or 1 Hertz for 2 weeks. We then stopped stimulating the animals and imaged them at the post-rTMS time point. We were very interested in finding out what happens after the cessation of stimulation. And another advantage of performing animal studies is that the animals are always able to come back for follow-up time points, which is something that's quite challenging in human studies. So we also imaged the animals at the week 1 and week 2 follow-up time points. In the 10 Hertz group, we found that connectivity to the basal ganglial network increased after 2 weeks of stimulation. These changes were seen in the contralateral somatosensory cortex, striatum, and hippocampus. This is really interesting because, as you might remember, the magnetic field of the coil that I use only reaches the stimulated hemisphere, as the human coil does in humans. But we know from human rTMS studies that rTMS can induce changes both locally and in remote brain regions. So after cessation of stimulation, functional connectivity gradually decreased over a period of 2 weeks and was back to baseline levels by the week 2 follow-up time point. In the 1 Hz group, we've actually found a decrease in functional connectivity in the prelimbic cortex after 2 weeks of stimulation. The functional connectivity was still lower than baseline after 1 week of no stimulation, but went back to baseline levels after 2 weeks. So in summary, from this study, what we saw was that there was an increase in functional connectivity following 10 Hz stimulation and a decrease following 1 Hz. Importantly, there were more widespread changes when we stimulated the rats with 10 Hz than with 1 Hz stimulation. These changes that we saw was actually similar to what we see in humans following brain stimulation. So next, we wanted to look at an animal model of depression. The model that we chose is called the chronic restraint stress model. It involves placing the rats in individual transparent acrylic tubes to restrict their motion for 2.5 hours daily for 13 days. The continuous and predictable nature of this stress is designed to mimic the everyday stress that people experience, such as a stressful job and familial stresses. Studies have shown that following restraint, the animals exhibit similar genetic and protein changes that are seen in human depression. But what about the functional, chemical, and structural brain changes that we see in humans with depression? Before we test the effects of rTMS on this model, we wanted to validate this model based on MRI-related changes. So we imaged the animals before and after the restraint procedure. We looked for changes in functional connectivity to the cingulate cortex using resting state functional MRI. As you have heard before, the cingulate cortex is a critical element of the default mode network and has been extensively studied in the context of depression. This network has been implicated in rumination, self-referential functions, and episodic memory retrieval. What we found was an increase in functional connectivity of the cingulate cortex to several brain regions highlighted here. The result of the current study is consistent with human studies and indicates an increased emotional processing at the cost of executive function in depression. We also acquired magnetic resonance spectroscopy data within the sensory motor cortex highlighted in blue. We found a significant decrease in both glutamine and glutamate levels following restraint. This is in accordance with previous preclinical and clinical studies, which have proposed that altered glutamatergic neurotransmission plays a pivotal role in the pathogenesis of mood disorders. Structural changes in the brain were detected using T2-weighted anatomical images. The hippocampus highlighted in red was extracted, and we found a significant decrease in hippocampal volume following restraint. A reduction in hippocampal volume has also been consistently associated with depression in humans. Diffusion MRI was also used to investigate structural changes in the brain within the white matter. We found an increase in fiber cross-section in both healthy and restrained animals, which indicates brain development and repair. So the colored regions that you see indicate an increase in the parameter being tested. Previous animal studies have shown that cerebral development occurs in rats until they're about 6 months old. Our animals were around 8 weeks old at the time of the baseline scan, so it would make sense that they would still be exhibiting brain maturation-related changes. However, the healthy animals show a considerably greater increase in fiber cross-section and also an increase in apparent fiber density, as you can see here. So for example, the healthy control showed an increase in fiber cross-section in voxels adding up to 81 millimeter cube, but the restrained animals only showed an increase in 8 millimeter cube. Therefore, animals subjected to restraint exhibit less brain maturation-related changes. This is a really important finding because it shows that even with a short duration of stress in the young developing brain, can have long-term consequences. In summary, we found functional, chemical, and structural changes in the rat brain following restraint. Importantly, these results are similar to MRI-related changes that we observe in human depression. This validates the rat model of depression Validating this model was really important because animal models are an indispensable tool for studying the etiology, progression, and treatment of depression. But there's still controversy regarding the validity of using rodent models of human neuropsychiatric conditions because the behavioral tests that we tend to use in animals are not directly comparable to changes in behavior in human depression. MRI findings, on the other hand, are more directly comparable. So the next step was to look at the effect of rTMS on this model. So since we have previously shown that 1 Hz stimulation has milder effects than 10 Hz, we decided to only use 10 Hz stimulation for the next experiments to investigate their effects in more detail in the depression model. In the clinics, rTMS is usually delivered once daily for 4 to 6 weeks, but there's increasing evidence that receiving multiple stimulation sessions per day can increase the efficacy of treatment while also decreasing the treatment period. So we conducted a small pilot study comparing the effects of this standard protocol to an accelerated TMS protocol in which the animals received 3 stimulation sessions per day for 2 weeks. We found that there were no changes in behavior in animals receiving the standard protocol, while animals receiving the accelerated protocol showed significant improvement in depression-like behaviors. Therefore, only accelerated TMS was delivered in our next experiments. So we performed the restraint procedure as before. After restraint, animals received an accelerated protocol of rTMS. After these 2 weeks of stimulation, MRI data was acquired at the post-rTMS time point. And we also then stopped stimulating the animals and imaged them at the week 1 and week 2 follow-up time points as with the previous study. So we had 3 animal groups. In the active group, the animals received active stimulation after restraint. In the sham group, the animals received a sham procedure during which the coil was just disconnected. The sham group was used to control for the effect of handling during the rTMS procedure. We also had a restraint control group, which underwent restraint, but no extra handling. So in order to determine the effects of rTMS in this model, we compared the post-restraint time point to the post-rTMS and week 1 and week 2 follow-up time points. Just a quick note that even though the sham and restraint control animals did not actually receive TMS, I will still be referring to the imaging time points as post-rTMS and so on. So when we compared the different time points, we were quite disappointed to find that there were no changes in functional connectivity, neurometabolite levels, or hippocampal volume after correcting for multiple comparisons. This was quite unexpected, but there were issues which I'm not going to go into right now. For the invasive measure, we wanted to investigate neurogenesis in the hippocampus. So we injected the rats with the DNA analog EDU at the mid-rTMS time point. At the end of the experiments, the animals were euthanized and the brain sections were stained for EDU. So the green parts that you see here is the dentate gyrus within the hippocampus. And this is where we expect to see neurogenesis. New cells in red are stained for EDU. There were also no changes in hippocampal neurogenesis in any groups. This makes sense because there were no changes in hippocampal volume either. So now let me tell you about the bit that did work. What we found was an increase in fiber cross-section in the diffusion MRI data. So this is what the results look like. There's a lot of images here, but I'll walk you through it. The findings, the first row shows the changes in the active group, the second in the sham group, and the third in the restraint control group. We can see that fiber cross-section increased with time in all groups when we compare the post-restraint to the week 2 follow-up time point. However, there was a greater increase in fiber cross-section in the active group compared to the sham or restraint control group. This is in line with previous animal studies showing that high-frequency rTMS can have a neuroprotective effect and prevent neuronal loss. Next, we wanted to investigate the white matter changes further using invasive methods. Immunohistochemistry was performed on a subset of the animals' brain slices to determine the level of myelination within the corpus callosum. These histological images are coronal slices of a rat brain. The red bit here is stained for myelin and is the corpus callosum. We found that animals receiving active stimulation had significantly higher level of myelin in the corpus callosum. The increased myelination could be one of the factors leading to the increased fiber cross-section observed in the diffusion MRI data. Low-intensity magnetic stimulation, as used in the present study, has been shown to increase survival of newly generated oligodendrocytes in mice, which may underpin the increased myelination observed in these experiments. The use of non-invasive imaging methods to investigate the effects of low-intensity rTMS in rodents allows us to directly compare rodent and human studies. We can also perform post-mortem studies like we have seen here in animals and link these findings to the rTMS-induced changes in MRI measures. In summary, this will help deepen our understanding about the effects of rTMS in neuropsychiatric disorders. We can then perform these studies in different animal groups to drive personalized rTMS treatment in humans. Thank you.